Welcome to the course on microbial risk assessment. In this course, we are going to cover the theory of microbial risk assessment and the applications of microbial risk assessment with software. Chapter 1. The fundamental theory of microbial risk assessment. Microbial risk assessment is a scientifically structured process consisting of the following steps: hazard identification, exposure assessment, hazard characterization, and risk characterization. Briefly, the hazard identification is the process of identifying potential hazards. Exposure assessment is the process of evaluating the level of human exposure to the hazard from the ingestion of food according to quantitative or qualitative analysis. Hazard characterization is the process of quantitatively or qualitatively calculating the level of human exposure allowance based on animal or human data. And lastly, risk characterization is the process of calculating risk with a simulation model based on the exposure scenario and hazard characterization. Microbial risk assessment is aimed at identifying health problems derived from the hazards in food. It is also aimed at providing a method to prevent and reduce hazards, establishing the standards to control the hazards, and evaluating the adequacy of the standards for the hazards. To understand the risk assessment, you should know the difference between a hazard and a risk. A hazard is a biological, chemical, or physical agent or condition of food with the potential to cause an adverse health effect. Risk is a function of the probability of an adverse health effect and the severity of that effect, consequential to a hazard in food. Risk is found by multiplying hazard with the exposure frequency. Now, let's study each step of microbial risk assessment in a little more detail. In hazard identification, a literature review is conducted to identify the potential hazards. Through this process, we search the physiological and biochemical characteristics of the foodborne pathogens that are being evaluated. We want to find any country that had foodborne outbreaks related to the pathogens and which food or foods are concerned. We will search for any domestic outbreaks caused by the pathogens in a specific food. Finally, we will look for the domestic and international standards to control the pathogens in specific foods. During the exposure assessment, we use quantitative or qualitative analyses to evaluate the level of human exposure to the hazards from the ingestion of food. In this assessment, food samples are collected to enumerate the initial contamination levels of foodborne pathogens and the contamination levels during the transportation of food. Exposure assessment is very important in microbial risk assessment, and within exposure assessment, developing a predictive model is the most important step. During food storage and transportation, the level of microorganisms in food vary continuously under constantly changing conditions like temperature, time, and so on. Thus, a predictive model needs to be developed to describe the fates of microorganism in food. The fates of microorganism mean decrease, increase, or no changes in the cell counts of microorganism. To predict the fates of the pathogens, primary and secondary models need to be developed. The primary model is for describing the changes in microbial cell counts as time changes. From the primary model, growth rate, lag phase duration, initial cell count levels, and the maximum level of cell counts are all calculated. The secondary model is for describing the effect of environmental factors on the parameters like growth rate and lag phase duration. They are calculated from primary modeling. The detailed procedures of developing predictive models will be covered in a later chapter. For the exposure assessment, the temperature and storage time of the food during the storage, transportation, retail, home phases need to be collected. These data are then inserted into the developed models to predict the fate of microorganisms. In addition, the data for the amount of food and the frequency of consumption per person have to be collected. Consumption amounts indicate the volume of food eaten by a person per time, and the consumption frequency is how often a person eats the food. Hazard characterization is the process of using a dose-response model to describe the relationship between the level of microbial exposure and the likelihood of the occurrence of an adverse consequence. At this stage, a dose-response model is used which quantitatively suggests the risk of illnesses that can occur as a result of ingesting foodborne pathogens. The dose-response model is influenced by the number of ingested pathogenic microorganisms, health and immune status of the human, the pathogenesis mechanism, and the food matrix. Common dose-response models include exponential, beta-Poisson, and Weibull-Gamma. The most suitable model is selected through the literature research. Risk characterization is the process creating an exposure scenario and hazard characterization in order to build a simulation model to calculate risk. To conduct risk characterization, an exposure scenario needs to be prepared by considering contaminants, pathways of exposure, concentrations of microorganisms, transportation and storage environments, and exposed populations. Under the exposure scenario, the fates of changes in the cell counts of the pathogens are predicted with the developed predictive model in exposure assessment. The exposure scenario is a series of data collected from monitoring the initial contamination levels, temperature, time, predictive model. It also includes the consumption data and the dose-response model. For simulating a scenario, probabilistic distributions should be determined for monitoring, temperature, time, and consumption data. Randomly selected data from a probabilistic distribution of the monitoring data is used as the initial level of the pathogens in the predictive model. The changes in the levels of the pathogens are simulated by randomly selecting temperature and time from the probabilistic distributions in each step. Subsequently, the final levels of the pathogens are predicted, and the data are combined with data from the probabilistic distributions of the consumption data. Eventually, the probability of foodborne illness is calculated with the dose-response model. The simulation model can be prepared with various software programs such as At-Risk, Crystal Ball, MATLAB, Model Risk, etc. If the simulation model is prepared with At-Risk, which uses an Excel base, it will look like the picture on the left. After running the simulation model, the probability of foodborne illness is calculated. Using the correlation coefficient, the relative influence levels of the input information on foodborne illness can be identified. It will show the influencing factors on risk.